Glucose, the main product of carbohydrate digestion in the small intestine, is actively transported through the mucosa into the bloodstream. It has been well established that the transport of substances against a concentration or electrochemical gradients involve a highly specific process that requires energy. This energy is usually supplied in the form of ATP. Thus, the process is highly sensitive towards chemical inhibitors that are capable of blocking any energy-producing cellular reaction. Active transport also demonstrates competitive inhibition and saturation characteristic and is thought to be carrier mediated. Active transport can be demonstrated by using sacs of the small intestine, which is filled with glucose solution and immersed in the same solution at the same concentration. The concentration of glucose inside the cell will increase with time if the glucose is being actively transported.
In this experiment, the first objective is to study the active transport of glucose in the small intestine. The second objective is to study the importance of oxygen in the active transport. The third one is to study the effect of 2,4-DNP to the active transport, and the last one is to study the effect of fluorescein to the active transport.
Hi, then we'll be moving to the procedures. The first step, you'll be supplied with 4 pieces of small intestines, 5 centimeters in length. This will help be inverted so that the mucosa surface is on the outside and serosa on the inside. Please handle the delicate mucosa surface with care. Next, place the intestinal pieces on filter paper that has been soaked in glucose ringer solutions. Then make a tight knot around one end of the intestine with a thread. The other end only require a loose knot. This will allow you to insert the solution later on. Then fill each sac with 1 ml of glucose ringer solution by slipping a blunt needle attached to a syringe through the loose end and tighten the knot as the needle is being withdrawn. Trim off any large end of tissue. Then measure 15 ml of the following glucose ringer solution into 25 ml test tubes. The first test tube contains glucose ringer plus oxygens. Second, glucose ringer plus 2,4-DNP plus oxygens. Third, glucose ringer plus fluorescein plus oxygens. Fourth, glucose ringer without oxygens. This is just for the constants. Okay, next, immerse one set into each test tube. Allow one end of the thread to hang over the opening of the tubes so it will be easier for you to pull out the sets. And then place the tubes in a water bath at 37 degrees Celsius. Next, the next step, prepare a set of polyethylene tubing from gas manifold and dip one end into bottom of each test tubes, test tube 1, 2, 3, so that a fine stream of gas bubbles run through the solutions. Leave all tubes incubate for 1 hour, 90 minutes if times allow. At the end of the incubation periods, remove the sets from the test tube by pulling on the threads using forceps. Blot the sets on the filter paper to remove excess fluids. And next, open one end of the sets and with care transfer the content into the clean small test tube. You may need to apply some pressure to remove the whole contents. This needs to be handled with very careful actions. Then, determine the glucose concentration in all 4 sets and the test tubes. Include the reading of the glucose ringer solution as controls. Thus, there will be a total of 9 samples for you to determine the glucose concentrations from. These are: first, oxygen set; second, oxygen tube; third, 2,4-DNP set; fourth, 2,4-DNP tube; fifth, fluorescein sets; next, fluorescein tubes; oxygen-free sets; oxygen-free tubes; and glucose ringer control.
The glucose concentration in test tube 7 is lower than that of test tube 8. Based on theory, the glucose concentration in test tube 7 should be slightly higher than that of test tube 8. Fluorozyme is a competitive inhibitor that will compete with glucose for the same binding site. So, the glucose that successfully bind with the transport protein still able to transport inside the intestine sac. The presence of fluorozyme will decrease the rate of transportation of glucose into intestine sac. The inaccurate of result may due to dilution of glucose solution when transfer in the test tube that being washed with water. The negative control is the control that being subjected to the same methodology without treatments. The test tube containing glucose stringer solution without intestine sac was also incubated in water bath. The test tube is a control and can be used to compare with other test tubes. According to the practical manual, the concentration of glucose stringer solution should be 180 mg per 100 milliliter. But in our experiment, the concentration of glucose stringer solution is 91.2 mg per 100 milliliter. This may due to dilution of glucose stringer solution during transferring to test tubes that being washed by water. The presence of little water in test tube may dilute the concentration of glucose stringer solution.
Based on result, we can observe the concentration of glucose in intestine sac in test tube 1 is higher than that of test tube 2. This can indicate the active transport of glucose into intestine sac at occur. But for test tube 3, the concentration of glucose is lower than that of concentration of test tube 4. So we can conclude that the oxygens are needed for active transport. Oxygen are needed in cellular respiration to generate ATP for process of active transport to be carried out. The glucose concentration in test tube 3 is lower than that of test tube 4. Based on theory, that should be equal concentration in both test tube. Without oxygen, the active transfer is inhibited and transportation of glucose will not occur. The inaccurate result may due to leakage of some glucose solution when transfer of glucose solution from intestine sac to test tube. For test tube 5, the glucose concentration is lower than that of test tube 6. This shows more glucose molecules are present in test tube 5. Based on theory, the glucose concentration should be the same for both test tubes because 2,4-dinitrophenol is a cellular metabolic poison that will inhibit the production of ATP. Without ATP, active transfer will be inhibited and no glucose transportation across mucosa. The inaccurate result may due to the dilution of glucose solution. The test tube that washed with water may have diluted the glucose solution.
Okay, let's talk about the discussion. In the gastrointestinal tract, glucose is transported across mucosa into the bloodstream by secondary active transport to distribute throughout the body. Glucose is needed for cellular respiration to synthesize ATP. The phosphorylation of ATP will increase the affinity of transport protein to bind with sodium ions. The binding of sodium ion will change the conformation of transport protein and expose sodium ions to the other side. Dephosphorylation will reduce the affinity of transport protein to bind with sodium ions. The sodium hence is released to the other side and establish sodium concentration gradient. The binding of sodium ions to co-transport protein will increase the affinity to bind with the glucose. The binding of glucose will result in the conformation change of co-transport protein and expose sodium ions and glucose to the other side. The release of sodium ions will decrease the affinity of co-transport protein to bind with glucose. The sodium ion move down the concentration gradient while glucose move against. In the secondary active transport of glucose, the ATP are needed for the process to carry out. The presence of 2,4-dinitrophenol will inhibit the production of ATP. 2,4-dinitrophenol is a cellular metabolic poison that can lead to rapid consumption of energy without generating of ATP. Secondary active transport will be inhibited without ATP. Fluorescein is a competitive inhibitor. It will compete the same binding site with glucose, hence the rate of transport of glucose will be reduced.
For conclusion part, the oxygen is used in cellular respiration to produce ATP. ATP is used in active transport to transport glucose from mucosa into the bloodstream in gastrointestinal tract.
2,4-nitrofuran is a metabolic toxin that will inhibit the production of ATP and prevent the active transport to be carried out.
Fluorescein is a competitive inhibitor that compete the binding site with glucose and reduce the rate of transportation of glucose. Thanks for watching our video.
Thank you.